Check it out now, y'all.
nanoHUB-U, online instruction.
Hi, and welcome back to Lecture 3 of Dynamic Atomic Force Microscopy Methods. The last class we were talking about how to measure electrostatic forces using dynamic AFM methods. In particular, we talked last time about the fact that you can go beyond Kelvin probe force microscopy and use single pass and two pass electrostatic force microscopy techniques. And we also talked about the fact that at the end of the day, when one acquires data using whichever EFM mode one is looking at, to really convert what one observes into local properties, you need to have a good model for tip-sample interaction, which gets a little more complicated than all the interaction forces that we have been talking about in part 1 of the class, which was focused on the interaction between the closest part of the tip of the AFM and the surface. In electrostatic force microscopy, in contrast, one needs to consider not just the proximal end of the AFM tip in coming up with interaction force models, but one needs to consider the entire geometry of the tip, and very often the geometry of the cantilever itself, which leads to much more complicated expressions for that relate the geometry of the cantilever to the forces being experienced and being measured. What we'll talk about today is magnetic force microscopy and how to measure magnetic forces. Much of what you're going to see in understanding magnetic force microscopy is going to be derived or based on similar principles as electrostatic force microscopy, except there are a few interesting specialized twists or specialized things that need to be taken into account while doing magnetic force microscopy. Again, this is not going to be a detailed exposition of magnetic force microscopy. It is simply providing an overview from a dynamic AFM method, or the role of dynamic AFM in understanding magnetic force microscopy images. There are a bunch of references at the end in the appendix, and I encourage you to go over them to understand more about the physics of magnetic force microscopy. So, what does magnetic force microscopy really consist of? What one is trying to do with an atomic force microscope is to measure stray magnetic fields that come out of a magnetic media or a magnetic sample. So, for example, shown here in the top left is a media. This could be a patterned data storage media. It could be a magnetic film that's been patterned in some way. What one has in such structures or in such samples are micromagnetic patterns. There are domains and there are magnetic moments associated with each of these domains, and they create a magnetic field above the sample. And the magnetic fields have gradients associated with them. And so when an AFM tip is brought at a certain height above from the surface, it experiences these gradients. We've already learned how force gradients can be sensed using dynamic atomic force microscopy, those are typically sensed using changes in amplitude or changes in phase or changes in frequency. Now, the point I want to emphasize, there are 2 points I want to emphasize here. What is different about the force gradients generated by magnetic samples compared to electrostatic or other samples is the fact that the gradients need not be all the time attractive, like in the case of electrostatics, or all the time repulsive. They can actually change depending on the interactions between the tip and the sample. The other thing I want to emphasize is that magnetic force microscopy refers to the particular case where the tip has a permanent magnetic moment. In other words, it's basically a permanent magnet at the end of the tip. And so the idea is you have these magnetic fields, let's say denoted by the vector H, and the tip comes in interacts with this magnetic field H, you need, of course, very good models that allow you to couple or understand the connection between the force applied on the tip and this magnetic field H. But the second important thing about magnetic force microscopy is that even if one is able to back out the force gradients that cause the shift in frequency, it turns out that there is not a unique one-to-one relationship between the measured magnetic field and the actual micromagnetic pattern on the surface. It turns out there are multiple patterns that can actually give you the same frequency shift or force gradients. And this is, again, something that needs to be taken into account, and which is why when one deals with magnetic force microscopy, modeling the interaction of the tip with the magnetic fields and understanding the connection between the structure of the media and the magnetic fields created by it are essential to interpreting the whole image. Now, in this particular lecture today, we're not going to go into details of how to model this interaction. This is provided again in much more detail in the references at the end in the appendix. I would also finally like to point out that magnetic force microscopy was perhaps one of the earliest implementations of dynamic AFM that have been known. In fact, the very first papers that came out on this subject were in 1987, 1 year after the invention of the atomic force microscope. So that's something to keep in mind. So what are some of the interesting specifics about how magnetic force microscopy works? Well, first of all, as I mentioned to you, we need to have a permanent magnet at the end of an AFM tip. These days, this is done by coating a ferromagnetic material, a permanent magnet effectively, at the end of a tip. This is usually a combination of cobalt or chromium, sometimes nickel iron tips. And you can purchase these MFM tips from many vendors off the shelf, and you can actually get them with different amounts of permanent magnetic moment associated with each tip. So you can get them in low, medium, or high values of magnetic moment or coercivity of the tip. Now, the reason why this choice is provided is because, as most of you know, when you've got data stored on a magnetic medium, or magnetic patterns written on a medium, if you apply a very large magnetic field, you can actually affect the very sample that you're looking at. If you've ever brought a magnet in front of a magnetic tape, you would have realized that you can actually erase the magnetic patterns. So in a similar fashion, what you ideally want to use is a tip that does not perturb magnetically or the magnetic domains or the magnetic moments of the sample that one is trying to image. So you want to have low coercivity, low magnetic moment tips. On the other hand, the interaction forces and the force gradients depend on the magnetic moment of the tip. So if you have very small magnetic moments of the tip, you get smaller forces and smaller gradients, and therefore you don't see much. So the choice of an MFM tip involves an interesting compromise between wanting a high signal-to-noise ratio, but at the same time not wanting to actually disturb the magnetic patterns of the sample one is trying to image. So the tip magnetic moment needs to be chosen depending on the softness of the magnetic material of the sample, the magnetic softness. The second interesting feature of magnetic force microscopy is that it's important to separate the topography of the sample from the magnetic force information. And this is done in two ways. This is done by taking a topography image in the first scan using either contact mode AFM or using tapping mode AFM in the repulsive regime, or even frequency modulated AFM in the repulsive regime. This has the effect of making sure that the topography image is acquired in a mode where most of the interaction forces actually come from contact, van der Waals forces, or from contact elastic forces, and very little contribution comes from the magnetic side. As a result, you can expect the topography image, if done properly, not to have information about the magnetic interactions. On the other hand, when you lift up off the surface, so let's say you've gotten a topography image using your first scan, you would lift off and move the Z piezo actuator, as shown on the right here, in a manner to mimic the measured topography of the sample. And so you're following effectively the cantilever now at a fixed height from the sample, because you're basically offsetting your Z motion by a fixed distance from the measured topography of the sample. Now what this allows you to do is the forces you end up measuring at an offset distance must be longer range forces, which must arise from magnetic interactions. So magnetic force microscopy is typically done using this two-pass technique, where a topography is first measured and effort is put into making sure that magnetic information doesn't reside in the topography information, and the second pass at a certain offset can be used to detect the force gradients. Now, the question is, what do you measure? What are the observables when one does the second pass? And this depends. There are many studies using many different techniques. One, you could do the second pass with the phase lock loop turned on, and you may want to do that in situations where you want extremely high sensitivity and you want to detect very slight changes in resonance frequency due to magnetic interactions. If you use a phase lock loop and do the second pass, you have to keep in mind that the second pass, there is no feedback on Z involved in it. Z follows a certain pattern as you move up and down. As a result, the observable in that case becomes the frequency shift. Remember, we're not using the frequency shift here to do a Z topography feedback. Rather, we simply measure the frequency shift at a certain fixed height above the topography from the sample at every point in the sample. So in that case, the observable will be frequency shift, and often frequency shift can be acquired at different Z distances from the substrate or at different liftoff distances. One can also do the same second pass measurement without a PLL, and if one does that, the amplitude and phase of oscillation are going to change because the excitation frequency is fixed. The resonance curve is like that, so as the resonance frequency shifts, both the amplitude and phase are going to shift. And people choose to either plot the amplitude signal or the phase signal in the second pass. But nonetheless, when one looks at what are called MFM images are basically images of either frequency shift at a certain liftoff distance over the sample, or a phase of oscillation at a certain liftoff distance from the sample, or amplitude of oscillation at a certain liftoff distance from the sample, depending on whether or not a PLL is used in the second pass. And as I mentioned, it's really whenever you see whenever you see an image called an MFM image, it refers to these observables made in the second pass. So I'm going to show you some typical examples of data from MFM magnetic force microscopy. One of the early applications of magnetic force microscopy was in understanding the magnetic patterns on data storage media, magnetic hard disks, magnetic tapes, magneto-optical disks. This was an excellent example because hard disk drives, for example, are extremely flat. They're polished surfaces, coated surfaces, and so the topography is not likely to show too many features beyond scratches that come from normal wear and tear. And the magnetic media are actually slightly below the top surface, and so it's an ideal sample to try looking for magnetic contrasts. And so here is an example where the topography image has been acquired and shown on the left, and not surprisingly, it does not show any magnetic features on it, but at a certain liftoff distance, when the frequency shift is plotted, you actually see very clearly the bit patterns that are written on the hard disk media. I show here another early example where the notion of how the bit patterns written on a hard disk drive deteriorate upon application of a very strong magnetic field are studied. Again, this is a common observation that if one takes a hard disk drive or any magnetic media and applies a very strong magnetic field to it, one tends to destroy the magnetic information recorded on the sample. So to understand this using magnetic force microscopy was the goal of this particular paper that was published some time ago now. What you see in this column of images, this, by the way, is data taken using tapping mode AFM. The topography image is taken with tapping mode AFM at a certain set point, and then there's been a liftoff distance. And the liftoff distance in this case has been somewhere between 15 to 20 nanometers all the way to 150, depending on the kind of sample one is looking at. But once the second pass is done at a certain liftoff distance, following a certain height above the topography, in this particular case, the MFM images are actually the phase contrast images associated with the second pass, which means the second pass is actually performed without a phase lock loop. And what you see in the sequence of images going down from A through H is the phase contrast images taken at an offset height, and each of these panels corresponds to an increasing amount of external strong magnetic field applied to the sample. What one notices is that at small external applied fields, the magnetic bit patterns show phase contrast between where the pattern is and the surrounding media. But as one keeps applying larger and larger external magnetic fields, the structure of the magnetic domains is being rearranged quite a lot until finally at an extremely high magnetic field, there is no further phase contrast at the certain liftoff distance. So this provides a very interesting example of how you can use two-pass magnetic force microscopy to observe changes in magnetic structure, microstructure occurring over a sample due to other external effects. And finally, I'd like to show an example or demonstrate an example where magnetic force microscopy is being used towards applications in biology. These days there's a lot of interest in magnetic nanoparticles. These are particles that typically have diameters less than 50 nanometers or so and are being used for all sorts of applications, from labeling cells, tagging cells, cell sorting, to actually being used for novel cancer therapy methods and so on. So what's interesting about trying to do magnetic force microscopy about such particles is that when you're dealing with magnetic particles that are so small in size, it turns out that the magnetic moment inside them is not fixed, but flips back and forth just under, due to energy from the environment. At room temperature, they don't have a fixed moment anymore, and they flip back and forth. And in order to get a fixed magnetic moment out of these particles, one needs to apply an external field, either vertical or horizontal field, depending on the configuration of the system. And it's only after one does that that one can hope to get a signal in the MFM image. So here I show an example from an article, the reference is shown at the bottom, where one is looking at magnetic nanoparticles, roughly diameter 20 nanometers or less, on a substrate. The top row corresponds to topography images taken in the tapping mode. The rightmost column corresponds to the images taken with without an external field, magnetic field. The middle column corresponds to images taken with an external magnetic field. As I said, the top row is topography. Underneath the top row are sequential images of phase contrast at different liftoff distances from the topography. The first phase contrast image that one sees is at 0 liftoff distance, and then 10, and then 20 nanometers, moving away from the sample. So I want you to focus attention on the middle and the second column. What's interesting about these images is that if you look at the rightmost column, we're looking at a magnetic tip, but no external magnetic field applied. You see the topography of the particles, But when one does the liftoff and measures phase contrast, remember, phase contrast is now going to tell us about the magnetic gradients. One notices that the phase contrast is hardly there as you lift off from the sample. So you see phase contrast when you're at imaging distance, but this is not surprising. We've already studied the origins of phase contrast due to dissipation and virial and so on. But as soon as one does the liftoff without the presence of an external magnetic field, one does not see phase contrast. On the other hand, if you go to the middle column where you have a magnetic tip and now you apply an external magnetic field, one finds that the phase contrast persists as one steps away from the sample, suggesting that the presence of an external magnetic field is essential to the MFM image formation. And this gives you, an idea of the fact that you can also use magnetic force microscopy to understand the changing magnetic properties of the sample based on external stimuli, in this case, external magnetic fields. So with that, I'd like to end this module on magnetic force microscopy. As I mentioned before to you, there are a number of references if you want to go into more detail that are provided in the appendix. Thank you.